This video is about mini-tubers. Mini-tubers is a model that we use in AP Bio to represent the 4 levels of protein folding. It uses this foam tube, which is why we call it a mini-tuber. And then it also uses push pins to represent amino acids. To remind you, a protein also goes by the name polypeptide. Poly meaning many, and peptide referring to the bonds that occur between amino acids. A protein is a polymer, meaning it is a long chain, and it is a long chain of units linked together, and those small individual units are called amino acids. In this exercise, we're going to use the tube and the push pins to represent a protein and then go through the 4 levels of protein folding. The first thing I'm going to do is start to put my push pins into my tube. Each push pin represents 1 amino acid. All organisms have proteins inside of their cells, and proteins do essentially every job inside of a cell. There are 20 different amino acids that can be found in proteins. Again, that leads scientists to believe that there is some relationship in terms of evolution between all different types of organisms. Because if all organisms have these same exact 20 amino acids, it leads us to believe that maybe there was once a common ancestor that relates all living organisms that are around today. Right now, I personally am randomly putting these push pins into order, but in a cell that would not be the case. The push pin represents an amino acid. The order of the amino acids is determined by DNA. So the whole function of DNA is to make proteins. In DNA, we have all those letters, A's, T's, C's, and G's, as I'm sure you remember from freshman bio. And those A's, T's, C's, and G's work in sets of 3 to determine an amino acid. More on that will be second semester. But right now, I have a chain of amino acids. That is the primary structure of a protein. So you're going to screenshot that. The order of amino acids is the primary structure, and that is dictated by the DNA. Then the secondary structure of protein folding is alpha helices and beta chains. An alpha helix is where the shape of the protein starts to coil. That's what a helix is. You probably have heard DNA is in the shape of a double helix, but proteins also can have a helical, like tornado whirlwind shape. Also in the secondary structure, there are beta sheets. Beta sheets are almost like zigzags. Most proteins would have hundreds, thousands, maybe even tens of thousands of amino acids. Mine is really quite small. But most proteins would have both a section that's a helix and a beta sheet. So you can take a screenshot of that. That's the secondary structure, alpha helix and beta sheet. The tertiary or 3rd level of structure is where the shape of the protein really starts to come into play. The shape of a protein is critical to allow it to do a certain function. We say in biology, shape is king, meaning the shape of a molecule allows it to do a particular job. The shape in which the protein folds is determined by these amino acids, which in turn, those are determined by DNA. So essentially the shape is determined by the DNA sequence. When the protein folds, it follows very specific rules based on the amino acids. There's 20 different types of amino acids, and they fall into a few big categories based on the way that they behave. Some of those amino acids have a negative charge. I am representing those here with red push pins. My red push pins are negatively charged. Some amino acids are positively charged. They are represented by my blue push pins. As I'm sure you know, opposites attract. So my negative amino acids want to be by positive amino acids. So here I'm pushing together a red and a blue. I'm pushing together a negative and a positive as best as I can. Up here I've got a blue, which is positive. It's going to find a negative to go by. Similarly down here, I'm trying to push my positives and negatives together. I am somewhat disrupting my shape here of the helices and beta sheets, but that's okay. So my positives are now all by negatives. My white push pins represent amino acids that are hydrophilic. Hydrophilic means that it does like or it does mix with water. The entire cell is made of a liquidy substance inside, which we call cytoplasm or cytosol. So it's very watery inside of the cell. Because of that, these white hydrophilic push pins are perfectly fine being exposed to the watery environment around this protein. However, my yellow push pins, which represent my hydrophobic amino acids, hydrophobic means it does not like and does not mix with water. My hydrophobic amino acids will all orient themselves to be facing inside the protein so that they're not exposed to the watery environment. So next I'm going to take all my yellows, which represent hydrophobic amino acids, and I'm going to twist them to face the inside of the protein. I'm still trying to maintain the alpha helices, the beta sheets, and the positive-negative connections that I've made. So I'm now twisting all my hydrophobic amino acids towards the center, the interior of the protein. My white hydrophilic amino acids are fine being exposed to the environment. And then lastly, for the third tertiary level, I have green push pins. My green push pins represent cysteines, which is an amino acid that tends to form a bond with itself. So the cysteines stay with the cysteines. So lastly, I'm just going to try to push my greens to be by other greens, my cysteines to be by other cysteines. So I've got a lot going on in the tertiary structure. I've got positives that need to pair with negatives. I've got hydrophilic amino acids that can be on the outside perimeter of the protein. I've got hydrophobic amino acids that have to orient themselves towards the center. And then we can snap a little screenshot of this, which is my tertiary structure of the protein. As you might assume, one wrong amino acid can really change the shape of this protein. If I were to have, let's say, a hydrophilic amino acid like this white one here, which is totally fine being on the outside of the protein, if there were a mutation in my DNA and the wrong letter got put in, and that caused the wrong amino acid to be put in, and let's say instead of that hydrophilic amino acid, I got a hydrophobic amino acid there. Now that guy has to be oriented towards the center of the protein. That now changes the shape of my protein. Similarly, if I had a positive amino acid and it got instead replaced by a negative, it would change the shape of which positives and negatives pair together. So shape is critical, and shape is determined in part by DNA. So when we talk about a mutation happening, like we did in the evolution unit, a mutation causes the protein to change. A protein that has a different shape might not be able to do a job, it might do a new job, or it might do a job differently. So that's why I might see different traits on organisms that have different proteins or different mutations. Lastly, to conclude, is the quaternary structure of protein folding. The quaternary or fourth level of protein folding means that most proteins, or a lot of proteins, are not made of just one single chain. Instead, they are made of multiple chains. Each chain has its own shape. Unfortunately, I forgot to bring enough push pins home to do this one. But imagine this has push pins on it, indicating the amino acids. And this particular chain is all folded. This one is folded. And in the quaternary structure, we have multiple proteins that come together to form like a megaprotein. So this is an example of quaternary structure. If you want to screenshot that, it is two or more chains all coming together. So that is the mini tuber exercise. The purpose of this is for you to understand the 4 levels of protein folding.